All right, welcome to E3 and E4 dot point. So the first one, we need to analyze and interpret data by calculating a mean, which I'm assuming you can do, and also calculating standard deviation. The second point is we need to work out what a test of significance is so that we can make comparisons between different sets of data. So when we do agriculture experiments, we get different results and different data, and we calculate a mean, first of all, an average, and then we also calculate other things like standard deviation, tells us more about the data. And so you can see here the mean is the same for these two curves. The one that is dotted has a higher variance, so the data is spread around further away from the mean, so that gives us a larger standard deviation, whereas the solid line here, the data points are much closer to the mean, and we call that smaller variance and a lower standard deviation. So basically, standard deviation is a way of quantifying the amount of variation or dispersion of data, and so a low standard deviation indicates that the data points tend to be close to the mean, while a high standard deviation indicates the data points are spread out over a wider range of values. You can use this chart here or a chart at the back of this worksheet to work out how to do standard deviation, but basically for these example numbers here, you need to be able to get this data, these results for standard deviation. In terms of test of significance, so basically when we get two sets of data or more, we need to compare them to work out whether they're significantly different. Now, if the data is too close to each other, we can't say that they're significantly different or they're not different enough, and we call that not significantly different. And we usually, in scientific experiments, we usually use what's called 95% confidence interval. So we work out, for each bit of data, we work out 95% of the data for this thing that we measured falls within this area. There's 5%, so there's 2.5% above and 2.5% below, and you've got the mean in the middle, and 95% of the data falls within this area. So what we do then is we can use this 95% and we can put bars around that. So if we looked at this random graph, we have the dots showing the mean on each thing that was measured, and the bars, they're also called error bars. Basically 95% of the data is within these bars. If it's been set to 95, we can make the bars show all the data if we want. We can make it show 100, but the problem is there might be anomalies, and 100% might make the bar up here. So for the sake of excluding anomalies, we use a technique where we cut out the 5% of anomalies, and we call this the 95%. So 95% confidence interval. So 95% of the data falls within this bar on this one, and the mean is here. Now, what we need to do is we look at these bars, and if the bars overlap, then we say that this one here is not significantly different from this one, because there's a chance that the mean could be here, and that the mean could be here, and they're actually basically the same. So because these bars overlap, these things are not significantly different. However, you can see that the end of the bar for this A here is actually not overlapping with C. So we would say that A and C are significantly different from each other, but because A and B overlap, A and B are not significantly different from each other. And B and C, because they overlap, are not significantly different from each other. However, because A and C bars do not overlap, we can say with 95% or more confidence that these A and C are actually different. Now, this is a little bit complex to understand, but it's important that you do, and it's important that you follow up on this if you don't understand it. So in terms of the answers, these are the answers to the questions. Some of them are just calculating means. I'll skip ahead just to show you the answers. Some are showing just calculating means. This one, the answer is standard deviation. This one here is A, 2012. 2012, there's a mean of 213 and a standard deviation of 7.87. Remember, you need to use the sample standard deviation, not the population standard deviation, which may be different from general maths. This is these two questions about the significance of what's the outline the need for a test of significance on data obtained from research. So tests of significance such as t-tests or ANOVAs. So ANOVA is an analysis of variance. T-tests are used to compare two means to each other, and ANOVAs are used to compare three or more means to each other, three or more sets of data. So these show whether the data is significantly different from each other, and they're usually based on 95% confidence interval. So 95% of the data from the two groups does not overlap, and it's said that therefore that they're significantly different. This allows conclusions to be drawn in research findings to illustrate outcomes of research. So I can